exactly noon, so I'm going to kick us off. I'm sure there'll be more people joining as time goes on, but this is just a little introduction to the cluster and then our seminar series, because this is the first one. And I know that we are probably all over the place, but I am, and the sort of cluster origins are on the traditional and ancestral and unceded territory of the Coast Salish people. So I wanted to make that land acknowledgement, including the territories of the Musqueam, Squamish, and Tsleil-Waututh nations. And we do thank them a lot for sharing their land with us. So the cluster itself, it's meant to be very inclusive, and I won't read all of those, our vision, but we do really want to be inclusive. And the reason why we started this cluster was because we don't know a lot about women's health specifically. And I'm just going to give you a tiny little bit of data because I think it's kind of shocking. So this is just in mice and rats, and this comes from data from 2010 to 2014. But you can see that males, the male-only studies are about 40%, and it hasn't changed that much. If anything, it's risen a little bit. But female-only studies, it's pretty steady. It's around 6%, so much lower. We, over the summer, some of the cluster members, the trainee members, and I did a study where we looked at both human and animal studies from 2009 to 2019. We got to write up the study, so it was a little bit of different journals because we focused on neuroscience and psychiatry journals, but you can see it's about 25% for male-only studies, and for female-only studies, only about 4 to 3%, so very few female-only studies. And I'd argue that we really do need these female-only studies, just like we need both sexes included. Big uptick, of course, in 2019 for both sexes. For today's presentation, feel free to hashtag it. If you want to ask a question, there's a Slido event code there, 612200, if you want to ask a question, because we've disabled the chat. We've done this because unfortunately women's health tends to get Zoom bombed, so that's why we have to put a number of security features on. Also tag us if you'd like to. You can join us, please, and receive our newsletter, or join us or like us on Facebook and Twitter. And lastly, we also have a Women's Brain Health virtual conference series. It starts next week. We're running monthly events. We have a number of sponsors that we couldn't put on these series in the conference without their sponsorship, so I thank them. And of course, always feel free to donate as well. Now, without further ado, I'd like to present. I'm going to stop sharing my screen here. Natasha Rajah. So while she's setting up her slides, I will introduce her. I'm really delighted. I met Natasha a few years ago. We both serve on something that she's very passionate about, Equity, Diversity, Inclusion Committee for the Canadian Association for Neuroscience. But really met in person, I would say, just since I think January or something, we met at a conference really in person. And since then, I can't even tell you how many times we've emailed and discussed back and forth. We've had a lot of discussions. So she's a kindred spirit. She received her PhD in 2003 at the University of Toronto, and then did her postdoc at the University of California in Berkeley. Lucky. In 2005, she started as assistant professor in the Department of Psychiatry at McGill University. She's now an associate professor there. But she also has an associate membership in the Department of Psychology, also at McGill. She's received CIHR New Investigator Awards. She has a Junior 2 Franz de Research Quebec Santé Health Research Scientist Award. I know that Santé is health. I know that my French pronunciation is not great. But as I said, she's an associate professor, and she's also the director of the Douglas Brain Imaging Center. In 2012, she was selected as one of Quebec Science's top 50 scientists under the age of 50. And more recently, in 2019, so just last year, she received the Women in Cognitive Science Canada Mentorship Award and the Haley D. Deblis Prize at McGill University for her work towards supporting equity and diversity at the Faculty of Medicine. See, I told you she really cared about that. She also is the one of the inaugural recipients of the CIHR Sex and Gender Chair in Neuroscience, Mental Health, and Addiction. She serves as editor-in-chief for the journal Aging, Neuropsychology, and Cognition. She's a senior editor for cognition and computational sections for the journal Brain Research. And she recently joined as an associated editor for Psychological Science. All of those are fantastic journals. I know that there are many other things I could say about her. I could talk about all of her, not just her awards, but all of her papers and fantastic research that she does. But she's going to tell you about her fantastic research. And I just want to give you one fun fact, because I think it's really great for us not just to hear about the accolades of people, but also about them as a person. So her one fun fact is that she's a massive sci-fi and fantasy fan. So am I. That's probably another reason why we get along so well. And her birthday happens to also fall on Star Trek Day. So, you know, how you can't get better than that. So thank you so much, Natasha, for being our first speaker, and I take it away.
Awesome. Well, I'm gonna minimize the faces so those don't get too distracted. So first, I wanted to thank Dr. Lisa Galea and everyone involved in organizing the Women's Health Seminar Series at UBC for inviting me to give a talk. I'm happy to meet everyone via Zoom and look forward to the questions at the end of the talk. So today I'll be talking to you about an exploratory analysis of sex differences that we did on a large-scale adult lifespan study that was conducted in my lab between 2012 and 2018. So in this talk, I'll give a brief overview, a background review of cognitive aging and episodic memory, touch upon some of the past work in my lab, and then present to you the Montreal adult lifespan study that was conducted in my lab, go over the planned analyses. So this study really was primarily focused on looking at aging and performance effects across the adult lifespan with a focus on episodic memory. And then talk to you about what the main topic is, which is the exploratory analyses and what we found about sex differences in brain aging and memory function. And I'll conclude by summarizing the findings from this study and then how that led to our current project and some details about our current project and some really fresh off the press preliminary data from that project. So before I go on, I just want to acknowledge everyone that's helped with this work. So here you see probably the last restaurant outing by the Rajah Lab in December 2019 for the holidays. And so here you have a lot of the trainees that contributed to the work that I'll be talking about today. So I want to thank them ahead of everything, say thank you for all the great work you do in the lab. I want to thank the funding agencies for the projects that have been done in my lab and also are currently ongoing, and the collaborators for both the adult lifespan study, which I'll be talking to you about today, and then the current project that is going on in my lab. So to begin, I always like to start my talks off with this graph that was taken from a review article conducted by Park and Ruta Lawrence back in 2019. Because it nicely summarizes the state of affairs in cognitive aging. So here's a graph showing Z-scores of performance on a variety of cognitive tasks. And as you can see, there's quite a lot of age-related decline in cognition. So with advanced age, you see performance declines with in speed of processing, so slowing, decreases in our ability of working memory and also long-term memory. However, it's not all bad news. There is some good news with age too. There is some stability of performance. So there's preservation of semantic memory and world knowledge. So in my lab over the last 20 years, I can't believe I'm saying that, we've been focusing on age-related declines in episodic memory. And the reason I've always loved deep affection for episodic memory is because it really is the cornerstone of defining who we are. So if we didn't remember our past, we really have little information or identity about who we are. And if we didn't remember our past and our interactions with other individuals, we'd have very low social connections. So episodic memory is truly integral to much of what we think of as being human. So I've been very interested over the years to understand what is episodic memory, what is the neural basis of episodic memory, and why does it decline with aging. So for those of you that maybe have not heard of the term episodic memory before, just a brief definition. So episodic memory was first defined by Endel Telving back in the early 70s, and he defined it as our ability to encode, store, and retrieve past personal experiences in rich spatial and temporal contextual details. So an episodic memory is a remembrance for a past event. Now that event contains both the content or the focus of your attention. And so when we're doing tasks like item memory or recognition memory tasks, you're basically trying to tap into your remembrance for past events and what was the focus of your attention. And that emphasizes your ability to encode, store, and retrieve intra-item associations. And then if you ask individuals about the how, where, and when of past events, you're tapping into the spatial temporal contextual details. So kind of the surrounding information of the past event. So this is memory for when you saw someone, where you last saw someone, and how, what was the context in which you saw them. And that type of memory, which I will refer to as a context memory, emphasizes inter-item associations and item context associations. Now, whereas memory for item or recognition memory emphasizes our mnemonic processes, I will argue that memory for context, the spatial and temporal context memory, emphasizes not only mnemonic processes, but cognitive control processes. So here's an example. So if there wasn't COVID and I was actually at UBC, you can imagine that this is me giving the talk in an auditorium at UBC. And so the next day, someone might ask you, oh, you know, what did you do yesterday? And you would say, oh, I went to this talk by Natasha, and she talked about aging and episodic memory. And so that would be the item or the content of your episodic memory for this talk I'm giving right here, this hypothetical world where COVID doesn't exist. Now, if you were then asked, you know, where were you in the audience when you saw Natasha give this talk? You really could answer that question by two means. So you could have a real deep mnemonic recollection using your spatial cognition to know exactly where in the audience you were seated when you were listening to me speak, or you could deduce that information. For example, you may remember that you were kind of looking leftward and that I was very far from you. So you deduce from that information that, oh, I was in the back right corner of the auditorium when I saw Natasha speak. So this just emphasizes that you could remember context information about an episodic event, both using mnemonic processes and what we refer to as cognitive control or strategic processes. Now, in healthy young adults, there's been a lot of research using brain imaging technologies to look at what are the neural correlates of episodic memory. And that work shows that episodic memory really relies on having an intact and functioning hippocampus and medial temporal lobe, but it also relies on having intact cognitive control mechanisms that include the prefrontal cortex and the parietal cortex. So it's not surprising that when we think about aging and age-related episodic memory decline, that these two regions are big players. Now, what I'll also show to you is that aging doesn't affect all aspects of episodic memory equally. So here's some behavioral data from two studies that were conducted earlier in my lab. So in this first study, we used verbal stimuli, and we asked individuals to encode words that were presented on the left or the right of a computer screen. And after they did encoding, they were asked to retrieve whether or not they recognized the word. So they'd see two words, one was old, one was new, and asked, which of these two words do you remember seeing? And that would be called a recognition memory task. And then on another task, they were shown two old words and asked which of these two words were seen most recently or least recently. And that would be a recency memory task that taps into temporal context. Now, in addition to those two memory tasks, we also had participants do what we call a working memory cognitive control task, where they saw two words and were asked to reverse alphabetize them. So in all these tasks, we had easy versions. So in the memory task, they saw 6 words if it was an easy version of the task, and 12 words if it was a hard version of the task. And in the cognitive control task, they were asked to either alphabetize 2 words that were very far apart in the alphabet, so it was easy to do that working memory manipulation, or 2 words that were very close to the alphabet. And so what you see here is the accuracy responses for young in blue and older in gray adults on these tasks. And you'll see that although aging is related to episodic memory decline, it appears as though memory for recency or temporal context is specifically sensitive to the effects of aging. Now, importantly, when you look at the control task, you see that a similar strong age effect is seen in the difficult working memory task, where the two words are very close together in the alphabet. So we took these results to imply that some of age-related deficits in episodic memory were most pronounced for context memory, in this case temporal context, and that some of that deficit may be related to cognitive control deficits in older adults, and not only related to mnemonic processing. Now, we did a follow-up study in 2010 where we looked a bit beyond temporal context memory, and we added a spatial context memory task. And we also included a different type of stimulus, which were faces. And that's because this study was done in Montreal, making use of verbal stimuli very tricky because you have all sorts of language abilities in our population. So we decided to use faces. And so in this phase, it was very similar to this, in this study, it was very similar to this one. So recognition, you were presented with faces, left or right, and asked to encode a bunch of faces, and then you had the decision of saying which of the two faces you see at retrieval do you remember seeing. The temporal task is identical to the recency task here, where you saw a series of faces again and asked to memorize the faces, and then you're presented with two old faces at retrieval and asked which of these two faces was seen most or least recently. And then the spatial context task was these faces were presented to the left or the right of computer screen, and at retrieval, you were presented with two faces, which were both old or previously encoded, and asked which of these two faces was originally on the left or originally on the right. And so what you see when you look at the accuracy data from this study is that in both young and older adults, you see a steady decline in accuracy performance, showing that recognition memory is the easiest to perform, followed by spatial context memory, and then finally temporal context memory. But what I'd like you to note is that in this particular study, there was no age-related deficit in item memory, but there were age-related deficits in spatial and temporal context memory. So this is just to show that it appears that source and context memory is more impacted by aging than item recognition, telling me that perhaps that's a good task to really investigate sex differences in brain aging and also risk factors for Alzheimer's in brain aging. But we'll get to that in a bit. So what were the brain mechanisms of this pattern of performance in young and older adults? So we had these individuals scanned while they were doing the retrieval portion of the study, so while they were actually remembering faces. And what you see here is a pattern of brain activation that correlated with performance in young adults. So increased activation in right dorsolateral prefrontal cortex in young adults, which is shown in bright blue and bright yellow, increased as a function of task difficulty. So the better they performed, the more they activated this region. And the more difficult the task, the more they activated this region. In contrast, older adults did not show a pattern of activation in this area for successful retrieval. And what you see when you look at the activation in that region in older adults is a flattening. So there really was no ramping up of activation with increasing task difficulty and retrieval success. So this implied to us that perhaps some of the age-related declines in context memory relative to item memory were due to deficits in right dorsolateral function. So we followed this up in this study by looking at the volume differences in right and left dorsolateral prefrontal cortex in young and older adults from this study. And what we found was older adults actually had a greater decrease in volume in right dorsolateral cortex compared to left, compared to young adults. And then what we did was we took the volumes of the right dorsolateral and the left dorsolateral as control, and we looked at how did the volume in these regions relate to activation at retrieval and task performance. And what we see here is activation maps from an fMRI study of transverse planes. So this is slices. This is the frontal lobes. These are the occipital lobes, and these are the temporal lobes. And this is just more ventral slice and going more dorsally. And what you see is in young adults, there is a correlation between volume of right dorsolateral prefrontal cortex. So the larger the volume of the right dorsolateral prefrontal cortex, the more activation you see in a more traditional retrieval network. So bilateral, lateral prefrontal activation, parietal cortex, and also some medial temporal lobe activation. And that was related to better performance in young adults. In older adults, we did not see that effect. So this implied to us that not only is it that there's decreased activation in this region, but that decreased activation may be due to underlying volumetric differences with age in this area, which then disrupts the network of brain regions engaged in episodic retrieval. So we followed that up by looking at another region of the brain that's important for episodic memory, which is the hippocampus. And so we did a volumetric segmentation of the hippocampus, and here's a sagittal section showing the longitudinal axis of the hippocampus, where we segmented the hippocampal tail, body, and head in young and older adults from this study. And what we found was, in general, there was volume reduction with advanced age, but that reduction was most marked in the hippocampal head, and it tended to be more right anterior hippocampus that showed a volume reduction with age. So we took the volume of that area and we looked at how does volumes in hippocampus relate to encoding activity in this same study. So what you're seeing here are activation maps, again, transverse slices, frontal, occipital, going from ventral areas to more dorsal areas, of regions that are more active at encoding and correlate with hippocampal head volume, and also correlated with memory success. And what you'll see is that you see activation in bilateral prefrontal cortex and in hippocampus and in temporal cortex and parietal cortex that increased with size of the hippocampus. So young adults that had larger hippocampal head volume showed greater activation in these networks at encoding, and that related to better accuracy on recognition, spatial, and temporal tasks. So the young are in the green and the older in the orange. And what you'll notice is in the older adults, you don't see that pattern. So in general, what we find from the studies that I've done in the past is that there are age-related declines in episodic memory. This seems to be more pronounced for context memory than for recognition. And this may be due to underlying differences in both structure and function of the medial temporal lobes and the right dorsolateral prefrontal cortex, which then disrupts the network of activations at both encoding and retrieval. So these were studies that were done looking at extreme age groups. So we know that by older age, so this is 65 and older, you see these episodic memory deficits. So this led me to ask the question, so when do these context memory deficits appear? Or when do they start in the adult lifespan? And then, not everyone performs this poorly with age. Indeed, there are some older adults that perform just as well as young adults on these context memory tasks. So what does compensation look like in the aging brain? So are there areas of the brain that increase in activation in older adults compared to young adults, and that also correlate with improved performance? So these are the two main questions that motivated us to conduct what I call the Montreal Adult Lifespan Study. And in this study, we focused on fMRI activation during episodic memory encoding and retrieval for both spatial and temporal contextual details. Okay, so this study was motivated to look at age effects and then age and performance effects. And so these were the planned analyses of these studies. So the sex differences that I'm going to talk to you about in a bit were really exploratory analyses. I want to highlight that because they weren't planned when we started the study, and that actually affected how well we could explore that question in the study. So here are the methods for this study. This study had two visits. So the first visit was a medical history screening. So individuals came in and they went through a bunch of neuropsychological data. And they had to basically meet some of our inclusion and exclusion criteria. So they all have to be right-handed, be self-reporting bilingual and multilingual since we're in Montreal. And then they were tested in their language of preference, so English or French. They also had to have no history of neurological insult, psychological illness, or substance abuse, so the traditional cutoff criteria for psychological studies. But importantly, for a subset of who I'm going to present to you right now, they could not have a family history of Alzheimer's disease or any other neurodegenerative diseases. So these were very healthy adults. They also had no history of diabetes, stroke, or heart attack, and they could not be suffering from high blood pressure, cholesterol concerns that were not controlled for 6 months. So they did neuropsychological testing, filled out a bunch of questionnaires, and then donated blood samples so we could get their ApoE genotype data that I'm not going to present right now because it's still in prep. And then they also performed some of our memory tasks in a mock scanner to make sure that they weren't claustrophobic and they could actually perform the tasks. So on the second visit, if they screened in, They came in for an fMRI testing session at the Douglas Brain Imaging Center. At that time, we had a Siemens Magneton Trio. It's since been upgraded to the Prisma Fit. And they came in and they had a structural MRI using the MPH sequence that allows us to do volumetry and cortical thickness analysis, and also functional bold scans to look at brain activity. Importantly, this was an event-related design, so we actually could look at brain activity when you're successfully encoding versus forgetting. And all of the analyses that I present to you from this data set is based on activations related to encoding success. So successfully encoding something that you subsequently remembered, and successful retrieval. So in the scanner, they did two types of memory tasks, a spatial and temporal context memory tasks, similar to the ones we've done in the past. And again, we had easy and difficult versions. So there were 4 tasks in total: spatial easy, spatial hard, temporal easy, and temporal hard. So here's a depiction of the event-related fMRI design. Don't know what that square is doing in the middle of that screen, but there it is. So during easy tasks, individuals were shown black and white photographs of age-variant faces, 6 of them, and these faces were presented to the left or the right of the computer screen, and individuals were told to encode the face and its spatial location for the spatial for the spatial context memory task, and also respond whether or not they found the face pleasant or neutral. So the easy task had 6 of these faces, and the hard encoding task had 12 of these faces. So after the encoding phase, they saw, again, a reverse alphabetizing distraction task, and then they performed a retrieval task. So at retrieval, they saw 2 old faces, so it was always 2 faces they'd previously encoded. Now, when it was a spatial context memory task, they were asked which of these 2 faces was either on the left originally or on the right originally. When it was a temporal context memory task, they were shown 2 old faces and asked which of these 2 faces were seen most recently or least recently. So the task demands were balanced for both spatial and temporal context memory in this study. So they had 12 behavioral runs. Each one was around 9 minutes. So they were in the scanner for 2 hours, which, you know, thank God they were. In total, at the end of 2 hours, you had 72 encoding events and 36 retrieval events. And we only analyzed individuals that had at least a minimum of 8 events per event type in these analyses. So I'm not going to go into detail about how we analyze this data because it's quite complicated, but we used a technique called multivariate partial least squares analysis. So this is a data-driven method which is optimized to explain the relationship between 2 or more blocks or matrices. So in our studies, we looked at the 2 matrices that we were concerned with was task design or behavior and brain activation. So we were asking the question, what pattern of brain activation most strongly co-varies with a task design or with the behavioral performance of individuals across these tasks? So we call it a behavior PLS when we ask the question, how does task-related brain activity correlate with the behavioral vector of interest? Okay, so now I'm going to present to you just the planned analyses and briefly go about talking about the results we found from that, because it informs the results we see in the sex differences analyses that I'll talk to you at the end. So the planned analyses looked at age and performance effects on memory-related brain function. So here are the sample characteristics that were published in Cortex in 2017 by a former PhD student of mine, Elizabeth Ankudovich. So here you have the sample size. So we had 128 adults, young adults ranged in age between 19 and 39, middle-aged adults ranged in age between 40 and 59, and older adults ranged in age from 60 to 76. You have the mean age, the gender distribution, and this is something I want you to highlight, which limited our sex differences exploration. The majority of our participants were female. And then you have the mean education, CVLT, which is a neuropsychological assessment of verbal memory. We have the free recall, the cued recall, and the recognition of these events. And what you see is that there's a typical age effect for free recall, which is similar to difficulty as context memory tasks, but there's no age effect for cued recall or recognition. And here you have the retrieval accuracy for the 4 tasks that they were doing in the scanner. So you have spatial easy, spatial hard, temporal easy, and temporal hard. And the ones with asterisks are the ones that showed an age effect. So we saw both an age effect and a difficulty effect, so an age by difficulty interaction, because the difficulty manipulation was successful more so for the temporal task than this spatial task with advanced age. But in general, you can look at the accuracy and you see that there's an age-related decline in accuracy for spatial hard, temporal easy, and temporal hard, and the parallel age-related increase in reaction time for all 4 tasks. So briefly, just to remind you, we did a behavior brain multivariate partial least squares analysis where we cross-correlated individuals' activation with a vector of age, so how old they were at the time of testing, and their performance for each of those types of tasks. And that gives us a cross-correlation matrix that you see here, for example. And then we apply what's called a singular value decomposition to that cross-correlation matrix to yield 3 values: a singular value that tells you how strong an effect you have, a singular image which tells you where the brain activations are seen in relation to a correlation profile. So it's very abstract now, but when I show you the results, you'll see what I mean by this. So the significance of PLS results were assessed by permutation and by bootstrapping. So in our analysis where we looked at age and performance, we first made sure that since age and performance are correlated with advanced age, we get decreases in accuracy and increases in reaction time, we decorrelated them. Basically, we regressed out the effect of age on performance to create 2 orthogonal vectors. And then we entered those 2 orthogonal vectors into our PLS analysis. And the first result we see is what's called an age-performance trade-off effect. So here you see a correlation of age and accuracy, and you see that they're going in opposite directions. So this means that the way that they relate to this brain activation pattern that comes out of that set of tasks differs, or in opposite direction. So with advanced age, you get increased activation in these red regions that include the temporal parietal junction, the anterior temporal pole, the ventral visual cortex, and the medial temporal lobes. And you get decreased activation in the midline cortical regions, including some fusiform areas and some parietal cortex regions with advanced age. But the relationship to performance is flipped, and that's why it's called an age-performance trade-off. So higher performance is actually related to increased activation in fusiform cortex, which isn't surprising, it's space stimuli, plus parietal and midline cortical regions. In contrast, poorer performance is related to activation in these midline medial temporal lobe areas, anterior temporal and temporal parietal junction. So that was the age-performance trade-off. In addition, we saw very complex patterns, which we called phase-related differences in brain behavior correlation. So here you see one of the effects that show a large pattern of activation in left frontal parietal cortex and right frontal parietal cortex. And these are areas of the brain that increased with age in activation at encoding, but did not predict subsequent memory. So these are areas that older adults engaged during encoding, but it didn't necessarily support their subsequent memory for faces. In contrast, activation of these same regions at retrieval does support memory. So it looks as though older adults are engaging these regions during a phase that is suboptimal task performance. And you see the parallel inverse effect here. So you see areas of the brain where increased activation at encoding, the red areas, support subsequent memory, but older adults do not seem to engage them in a preferential way. But you do see older adults engaging these areas at retrieval. But engaging these areas at retrieval is related to poorer performance. So perhaps some of the poorer performance that we see in older adults in advanced age is due to the misattribution of activating these areas at encoding instead of retrieval, and activating these areas at retrieval instead of encoding. Now, when we extract these areas of activation, we looked at how did they change or differ between age groups. Since this is not a longitudinal study, I don't use the word change. What you see here is that aging is not linear across all regions of cortex. So what you have here are activations of posterior occipital cortex and parahippocampal gyrus, parietal areas, and prefrontal areas. And this is the encoding-related activation in young, middle, and old, retrieval-related activation in young, middle, and old. And what you'll see is really the age effect in activation is flat at encoding and shows a linear effect here. So in these areas of the brain, there is a linear effect of aging, but in parietal, there doesn't seem to be a linear effect of aging, where you see an increase at encoding. At retrieval, older adults continue to show an increase in activation in these regions, but young and middle-aged adults don't. So if there was a linear effect, you'd expect that middle-aged adults might be up here somewhere. And similarly, at encoding for anterior prefrontal areas, you see a linear effect at encoding, and not so much of a linear effect at retrieval for these areas. So one of the critical things that we find in this study is that change occurs at midlife. So you see changes in brain activation at midlife for many areas, and performance also changes at midlife. So at midlife, you see it seems as though it's a critical period when episodic memory decline, as measured by context memory, first arises. And our between group activation analysis shows that age-related differences in task-related activation did not follow a linear pattern. So by midlife, you already see differences in prefrontal cortex and visual cortical activation, but only in later life do you see changes more in parietal cortex activation, suggesting that these changes are nonlinear with advanced age. And then you also see in the behavior PLS analysis that there is an age performance trade-off, and that some of the age-related differences that we see, such as increases at encoding, were not related to better subsequent memory. So remember, we started the study trying to look for compensation in the aging brain. And our initial analyses, where we do not consider sex differences, we failed to see any strong evidence of that. So we wrapped up the study looking at age and performance, and then we became curious about the effect of sex or biological sex or gender on our brain activation patterns. So why did we become interested in sex gender effects in our study? So at the same time, there was a lot of talk about how there's an increased prevalence of Alzheimer's disease in women compared to men. Now, interestingly, episodic memory is one of the cognitive features that are most impacted by Alzheimer's disease, and also shows some of the earliest changes as a sign of Alzheimer's disease. So here you have some data from an early study, a behavioral study by Bachman et al. It was a longitudinal study where they looked at adults and they followed them 3 and 6 years to see whether or not how their memory changed over time. And so here you have older adults that later converted to Alzheimer's disease and older adults that did not. Now what you'll notice is there's free recall at time 1 versus free recall at time 2, recognition memory at time 1 versus recognition memory at time 2. And what you'll notice is that in older adults with Alzheimer's disease, they were already showing episodic memory deficits 3 to 6 years prior to the diagnosis. And this deficit was more marked for the free recall task compared to the recognition memory task. So this finding and many others has spurred a significant amount of research to look at biomarkers of AD-related memory decline in preclinical populations with risk factors for Alzheimer's disease. And take into consideration this and the fact that women are at greater risk for Alzheimer's disease compared to men, we thought, here we have an adult lifespan study that looks at episodic memory, and let's explore whether or not there is indeed a sex difference in episodic memory brain function in these women compared to men, which may also kind of hint at what may be going on in women who are at risk at Alzheimer's disease. So I would argue that even if there was no effect of sex on Alzheimer's disease risk, it's still important to explore sex differences in brain aging, because it could be that the neural basis of cognitive decline may not be the same. So even though behaviorally women and men might show similar behavioral patterns, the reasons for that decline may differ in women compared to men. And if there are compensatory mechanisms, which we did not see in our study that combined women and men, perhaps we do see them appear differentially for women compared to men. And that's important to know because it's important to know what areas of the brain remain intact and might actually support memory function in late life if it differs between women and men for developing new interventions and to support prevention. So we did a follow-up exploratory analysis where we looked at sex differences in brain aging and risk effects. So the reason for looking at this, this data in this way was to really try and understand first, can we use context memory tasks to identify early pathological changes in memory and brain function, and do they differ in the sexes? And do they also differ based on Alzheimer's disease risk factors? So we already discussed the results from these two papers, and now I'm going to talk to you a bit about results from the exploratory analyses that have been published. And so these are results from a student of mine, Savanya Subramanian, which was published in Journal of Cognitive Neuroscience in 2019. Here you'll see that our sample size is very different in this study compared to the adult lifespan. So the adult lifespan, we started with 128, and then we actually just completed the full analysis where there's 167 adults in that study. But to look at sex differences, we had to be a bit more careful. So as I told you, like a lot of the changes we observed in our study started at midlife. And as you know, midlife is also the time of menopause transition in women. So we really were mindful of how do we address this? How do we kind of, since we don't have much hormone data other than self-report menopausal data, we really couldn't say much about women's health in that way in this study. We decided to control for that by taking out women that had just reported converting to being postmenopause. So in this study, we only looked at middle-aged women that were in pre or perimenopause, and that's why they're a lot younger. And then we looked at women in older adults as postmenopausal women, but well into postmenopause, and young women that were premenopausal in this study. And our rate limiting factor are really the number of men in the study. So if you remember, 70% of our population were women. So what we decided to do was take the men that participate in this study and match our women on education and on age. So this, and the reason we did this is that education is known to influence memory function, and there's some argument that some of the sex differences in memory and brain aging might be due to differences in education. So we wanted to control for all that. And so we matched on all our neuropsychological variables as well. And so you can see that our two biological sexes of self-identified male or females in this study did not differ on any of our neuropsych measures, nor in smoking history or BMI, because this was something that we needed to control for as well. So in this study, because we're already looking at something complex like sex differences in the effect of brain aging, we decided to target only the activation results we had from the spatial context memory task. So here you see that we see the traditional effects of age-related decreases in retrieval accuracy, particularly for the spatial hard task. And so this is just the demographics of the group that we analyzed for the sex differences in the sample. And then we used the same methodology, so behavior PLS, where we looked at both age and accuracy and its relationship to brain activation during spatial context encoding and retrieval. So here's our first result from that behavior PLS analysis. So just to orient you, here are the patterns or correlation for age. So here are males and females, and here are the pattern of correlations for behavioral accuracy for males and females. So we had three significant effects from this analysis, what we call latent variable 1, 2, 3, and 4. And what I want you to note going across all of these is when you look at accuracy, the bottom two, they're pretty much similar across the three significant effects in women and men. And this implied to us that in general, the accuracy-related activations independent of age were similar in both women and men. So the way you would interpret this is here's a pattern of brain activation that came out from this analysis, and these areas are all blue. So they're positively correlated to negative correlations and negatively correlated to positive correlations. So what I'm saying is that increased accuracy, so increased subsequent memory effects in men and in women and at retrieval in women was related to greater activation in left frontal parietal cortex and right frontal parietal cortex, in addition to midline regions. Now, when you look at the age effects, what I want you to note is that in men, these regions seem to show similar activation across the entire lifespan. So there's no age-related correlation pattern to activations in these areas. So that's interesting because our previous results, we saw a strong age-related correlation with these areas of the brain. And so what this tells us is our previous results were driven by the women. And indeed, in women, we see an age-related increase in activation at encoding for these areas, and an age-related decrease in activation in these areas at retrieval. Now, importantly, age-related increases coding in women supported subsequent memory. So if you remember in our previous analysis where we did not disaggregate our data by sex, we did not see any evidence of compensation. But here, when we did disaggregate our data by sex, we do see some evidence that left frontal parietal and right frontal parietal activation at encoding supports subsequent memory, but this effect is specific to women with advanced age. Now, we also see some age effects in other areas of the brain where there are sex differences in activation patterns at encoding versus retrieval in women compared to men. So in women, increased activation in these left ventrolateral prefrontal areas and visual areas and midline cortical regions was related to poor performance. Now in men, they show the opposite age-related effect, but the resulting effect for behavior was the same. So this is an effect that shows age-related declines in both women and men, but the pattern of age-related decline differ in the two sexes. So what is causing the age-related decline in women is more related to these red regions, and the areas that are related to age-related decline in men are more these blue areas. So that's important to know if you're trying to identify some marker of, say, memory deficit risk for perhaps Alzheimer's disease or MCI, to know that women and men may manifest this differently. And we see the same continued sex difference here in the last effect that we found, where you see areas that show an age-related increase in activation are in red, related to poorer performance in women, and areas that related to age-related decrease in men are in blue. And at retrieval, they're related to better performance, but men do not activate these regions at retrieval. To summarize the sex differences analysis, which is quite complex because it includes this multivariate brain behavior associations, we found evidence for both sexual convergence and divergence in brain behavior associations. So women and men exhibit similar brain behavior associations at encoding and to some extent at retrieval. So the brain behavior link is similar in the two sexes, and behaviorally they were similar as well, but the age effects differed. So women and men exhibited different age-related activations during encoding and retrieval. In men, age-related increases and decreases in brain activity were in general related to poor performance. And in women, we saw evidence of compensation. So where age-related increases in left prefrontal cortex and parietal cortex at encoding predicted subsequent memory. So the conclusions from pretty much the adult lifespan study and the sex difference analysis of that study is first that midlife is a critical period of time where episodic memory deficits first arise, and these deficits seem to be related to initial differences in prefrontal and medial temporal and ventral visual activation, and late life differences may be related to parietal cortex activation. And the neural correlates of age-related spatial context memory decline seem to differ in men and women. In women, it appeared to be related differences in midline cortical regions, in addition to these more anterior prefrontal and lateral temporal areas. And in men, it really was more the temporal cortices that showed age-related decline and related to poorer memory with advanced age. And in addition, by considering sex, we were able to see evidence of age-related compensation in left lateral prefrontal cortex and parietal cortex in women only. So this highlights the importance of considering sex in the cognitive neuroscience of aging and memory. And this led us to ask, What about the effect of AD risk in women and men? So there's a lot of evidence, growing evidence, that women who have an ApoE4 allele may be at greater risk of developing Alzheimer's disease compared to men who have an ApoE4 allele. And so we collected blood on these individuals from the adult lifespan study. And so we decided to look at the behavioral data to begin with to explore whether or not there is an ApoE4 effect in women compared to men. So I'm going to present to you just the behavioral analysis because the activation analysis is still in preparation. So when we disaggregated our data by ApoE4 status, the truth is we didn't have that many ApoE4 individuals. We had 48 out of our total sample of 154 for this analysis. So 154 individuals agreed to give blood and were genotyped, and of those, 48 individuals had ApoE4 allele, and 12 were young, 21 were middle-aged, and 15 were older. And so we don't really have a large sample size, but we have enough samples for power analysis of our behavioral of our behavior, since episodic memory age effects can be observed with the power with a sample size of 9 to 10 in this particular task. So when we look at the behavioral analysis from this study, looking at considering ApoE4, sex and gender, and age, this is an interesting result. So what we see is when we just look at age and task effects in the sample within ApoE4 and ApoE4 negative adults, you see an effect of task. So there's a increasing difficulty from spatial easy to spatial hard to temporal easy to temporal hard, which I've already shown you. And this is also present in both E positive and E negative individuals. And the age effect is pronounced when you don't include sex and gender in the analysis. However, if you include sex and gender as a between group variable and not a covariate, but a variable of interest, you wipe out the age effect, but only in the E4 carriers. So why is that? So I want you to draw your attention to this bar graph. What you'll notice is that, sure, there's an age effect, or there's an effect of task difficulty. Is there really an age effect in the same linear way that we see in the E4 carriers in the plus E4 individuals? Indeed, what you notice is that the middle-aged and the older adults in the E4 carriers are performing the same. So this implies to us that in ApoE4 carriers, there seems to be a quadratic decline of episodic memory ability, where episodic memory ability deficits might arise earlier at midlife. But that midlife effect that we saw in the full lifespan analysis may be driven primarily by these E4 carriers. Whereas when you look at the minus E4 carriers, you see that the effect truly is more linear as what would be expected from an age effect. So this is really exciting to us. So we're now following this up with the activation analysis of this data with our full sample of 154. So stay tuned to see what we find with that. Okay, so based on those findings and the fact that midlife seems to be a critical period in adulthood, and midlife is also the time that women experience menopause, we've now taken all of those findings to start a new project in our lab. And this is the project that's ongoing. So we're conducting what we're calling the Women's Brain Health, Aging, Menopause, and Memory Study, or the W-BHAMM study. I'm always looking for an acronym. And in this study, we're going to examine how individual differences in sex hormone levels and ApoE genotype influence memory-related brain function in women and men. So we're going to focus on midlife in this study, since we think it's a critical period in adult development. And we have enrollment going on right now where we're focusing, targeting areas that midlife women might go, like the gyms and yoga studios. Of course, COVID has significantly affected our recruitment at this time, but our enrollment is through an online portal to make it a bit easier, followed by a visit 1 where they do neuropsychological testing and a blood draw. And then a second visit where if they meet our inclusion criteria for neuropsych, they come in, they give another blood draw, and they undergo a series of MRI scans. So in this study, we not only get T1 weighted and task fMRI scans, but we also get an ultra-high T2 weighted scan that allows us to look more deeply in the medial temporal lobes of these individuals. And we also get a 10-minute resting state fMRI scan that will help us to understand how intrinsic connectivity and activation in individuals may predict task performance, and relate to deficits in memory with advanced age, and as a factor of sex, hormone level, and ApoE genotype. So this study is ongoing, and we have recruited 80 individuals so far, 10 males, 34 premenopause young women, 20 pre-peri-middle-aged women, and 16 postmenopausal women. The collaborators on this study are my student Sivanya Subramanian-Pillai, my student Alicia Crystal, my student Alicia Duval, my student Mile Leparco, and Jamie Snight, and in addition to collaborators at University of Toronto and in the US as well. And here's the task fMRI design from this study, which is slightly different. So we decided to amp up the spatial context memory task and make it a true spatial context memory task where they have to remember not only the face, but its location in one of 4 quadrants. This is my daughter when she was 2 years old. So you had to encode a black and white photo of age variant faces presented in one of 4 quadrants. The easy task had 6 encoding stimuli, hard task has 12 encoding stimuli, so similar to my previous studies. You're asked to encode the face and its spatial location and evaluate its pleasantness. And then you have a break where you rate how well you memorized the stimuli. And I could answer any questions about why we changed this up a bit. And then after this break, you do a retrieval task. And this is different from our previous designs. So now we have only one face in the center of the screen, and you have a complex mapping of the face to response type. So when you see a face, half of them will be old, half of them will be new, and you'll be asked to say whether or not the face is familiar. Was it in the top left, the top right, the bottom right, or is it new? And the reason we decided to do this design is because it allows us to tap into whether or not you're using mnemonic processes or familiarity-based processes to do the task. So are you engaging more medial temporal lobe areas, or are you engaging more prefrontal areas? And it also allows us to differentiate familiarity-based retrieval from recollection-based retrieval, which is important when we're looking into ApoE risk and Alzheimer's risk, since Alzheimer's disease is thought to affect areas of the brain that are more sensitive to familiarity-based decisions earlier on. So what we would predict from the study is women that have an E4 allele might show increased response familiarity compared to true recollections, compared to their age-matched cohorts that do not have that E4 allele. So this design allows us to explore that. And then after retrieval, they do another break where they rate how well they think they did. So this is just some preliminary data from the women that we've tested so far. So this is just the women, the young, middle-aged, and middle-aged postmenopausal women. And what you'll notice is when you look at hit rate, which is both familiarity plus recollection of spatial context retrieval, you do see a menopause effect. So in this analysis, we did an age by menopause between group repeated measures and ANOVA, and we see a significant effect of menopause, but not a significant effect of age. So this effect seems to be driven by menopause as opposed to age, which is important to know since menopause and age correlate. So I'll conclude by saying we need participants. So if you're watching this Zoom from Montreal, please do try and participate in our study, since we really are desperate for middle-aged women because we're very busy at midlife and it's very hard to make the time to come up to participate in research. So again, my acknowledgement slide to say thank you to my trainees, my collaborators, and funding agencies, and thank you to Lisa for inviting me to give this talk. I hope it wasn't too difficult to follow. It's very surreal to give a Zoom talk.
It is. Well, we might have had a little bit more of a general audience, and there are some questions, but I'm going to take liberty and ask one question. I have two, but I'll try not to take too much. Thank you very much. You can use your little reactions as a clap so that we don't unmute everyone and then have all sorts of noise in the background. But thank you very much. That was really great, really interesting. And, you know, obviously I'm really excited to see the menopausal status data coming out. And also, you know, really striking that you don't see a lot of studies on sex differences and cognitive aging. I mean, I've looked myself and there are very few. So it's really great that you did that exploratory analyses. I'm wondering, do you have any, first of all, why didn't you look at the temporal? If it sort of follows the animal literature, which it might not be, I am pretty sure temporal has a lot to do with the CA1 region, which is one of the first regions to show neurodegeneration.
Pragmatic reason.
Yeah,
So,
okay.
so temporal context memory tests are very hard. And so when you do an fMRI study, you need enough correct events to analyze the hemodynamic response function. And you just have people flooring, so you don't get enough events.
I see.
You can't really get a good signal.
Right. Okay.
So we decided to drop it and make it.
That's a pretty good reason then. All right. So I'll just ask some of the other questions because otherwise it's not fair. Given that bilingualism, which is going to be a good issue that you have in Montreal, has been linked to cognitive advantages, do you think these can generalize to unilingual or people?
So everyone in our study is bilingual or multilingual. So that's a requirement for all studies in my lab just because it is in Montreal. We do look at the language variables. One of my students, Savanya, looked at bilingualism as a reserve factor in the adult lifespan behaviorally and has published that. Happy to share the publication for that. And what she found really motivated some of the work as well. So what she saw in that study where she looked just behaviorally to look at how does bilingual language and language usage really, so she did a deep dive into a language questionnaire to see different measures of language use and ability. And she looked at how that related to neuropsych assessments. So the hypothesis being that if you're bilingual, you'll have an advantage and you show higher executive function with advanced age. And what she found was that was only true for women at midlife.
Mm.
So women at midlife, if you're bilingual, then the bilingual advantage seems to support executive function to a greater degree. But we did not see that in the young adults and we did not see that in older age.
But that's sort of your target group, at least in terms of interventions.
So that's exciting that we saw that in midlife.
That's really interesting. So another question that's here that I'm going to morph my second question into as well is, did you see differences between left and right in the prefrontal cortex in women? I think you did. I think you saw most of it in left.
Most of the activation at encoding was at left that supported subsequent memory. So the compensatory activation. So in the aging literature, I had these slides. I deleted them because I was already afraid I was going to go too long. So there's a long history of aging literature that looks at what are the patterns of age-related compensation. And there's a hypothesis that with advanced age, you increase activation in prefrontal cortex, and that activation increases in bilaterality. So if a task is mostly using the right hemisphere in young adults, then it engages also the left hemisphere in older adults. And this is thought to be compensatory. And it's actually called the Herald model and was put forward by Roberta Cabeza. And so what we see here is actually in line with that. So with advanced age, you see greater left activation, right? And in addition to right, and that left activation, not the right, correlated with better performance in our older adults.
But only in the women, or is it in both?
Only in the women.
Yeah, only in women.
This is what was really astounding about the study. We were searching for compensation. Like the way this grant was actually framed was to look for compensation in the aging brain and what are the compensatory mechanisms at midlife and late life. And we saw nothing until we split by sexes.
I think that you saw a few things. I think once you split by sexes, that's a lesson to everyone. Split by the sexes. Yeah, that was really, really fascinating.
And I think historically, the reason people haven't done this in cognitive neuroscience in general and in cognitive neuroscience of aging is that behaviorally, there's no difference in the sex.
Right.
People assume, oh, there's no behavior difference, so there can't be a brain difference.
Right.
What we see is, the way that they go about doing the task and the reasons for the decline differ.
Yeah, it's that. I mean, and I think I can't show it. Obviously, I'm not going to show it now, but any better than Becker and Koob have a fantastic slide where they talk about the types of sex differences. So even if your behavioral effects are the same, you might see completely different neural mechanisms that are involved, and there's a lot of evidence to support that, including your own. Do you like any reason why? Because I think you saw more female only when in PFC right? And I've written something else down, parietal maybe? Parietal and PFC, and male was more temporal? Like when you did see
Yeah.
sex differences, is that true?
Well, that's what we saw. Like, I don't know if it's true. That's what we
Oh,
saw.
yeah, it was true from your data. Yeah.
It was that
Any,
true data. So let me see.
do you know any reasons why that might be true? Why males?
I have like a working hypothesis. I'm not sure if I should share because I think it's
Oh
kind
yeah,
of
sure. You don't have to share it. You can share it like 2 years from
it's
now.
kind of it's kind of I don't have any reason for believing this. I guess so. I know.
Those are my favorite.
But so one of the ideas I've been thinking about is so with women, so the regions that women use to support memory and that showed age-related decline were the medial, prefrontal, and limbic areas. And with advanced age to support memory, they kind of reached out to the lateral, prefrontal, and parietal cortices. And in men, they went from using those areas, the left parietal and prefrontal were similarly engaged across the age in men. But then they showed engagement of these more lateral temporal and superior temporal areas with advanced age. It didn't seem to be compensatory in them though. But one of the ideas, kind of like in this study, we did not control for for job use. So maybe some of these older women, maybe occupational history matters. And so men are more likely, especially in the older generations, to have had work. And so they engage these more cognitive control mechanisms, like that kind of idea.
Yeah,
and then that are able to engage them in late life maybe also had careers. So we really need to dive into career trajectories of these individuals, and is that what might be playing in? And if men are like with the temporal areas, this whole I don't like that, but this whole notion that women are more verbal than men. And so the idea that as you age and the systems that you're using or traditionally used start to fail, maybe you start to reach out into systems that aren't as strong. So if men are less verbal, maybe with advanced age, they start to engage this system that they don't really rely on a day-to-day function, but when things get hard, they start to rely on.
Really interesting.
Yeah.
All right, well, I'm mindful of the time, so I'll just say there are some more questions about parenthood, which is one of my favorite, if you notice any differences, and hysterectomy and oophorectomy. So in terms of parenthood, you could think of parity, this parity. We know that there's some work, as I know you know.
I know, Lisa.
But I'm sure that's going to be something that you'll look at in the future.
Yeah, so we went into this adult lifespan study to look at aging and performance, and then we ended up doing sex difference work, right? Now our study is really focused on sex differences. So we do look at parity, we do a reproductive health questionnaire, and we are going to have I'm really excited. So that's why I try and volunteer for the study so we could get this completed. And so it's a project that's funded for the next 4 years. And so it's, hopefully we have answers.
All right, well, we wish you good fortune finding those subjects so that we can learn more about cognitive aging between men and women and across menopausal status in women themselves. And
give a Zoom talk. I'll that way you only see your screen and yourself.
I know, it is very difficult, I know. But thank you, we really appreciate it. So thank you very much. And I want to make sure that we get off the call because I'm mindful that other people have things to do. I don't know what. I mean, I'd rather just talk to you. But anyway, thank you very much. Thank you, everyone, for tuning in. And we'll see you next couple weeks. Bye.